Viruses don't do many of the things that we associate with life, but what they do well is reproduce. They will take over the host cell and really turn the host cell into a virus-making machine. And so we're going to take a look at how viruses do this by looking at some examples of the viral reproductive life cycle. So there's some basic steps here that I've outlined: the attachment, entry, integration, synthesis of viral components, viral assembly, and viral release. And this is kind of the basic outline of a viral reproductive cycle, but there can be a lot of variation among the different types of viruses, and we'll also see that one virus can even have alternative reproductive cycles. So in order to understand the viral reproductive cycle, we're really going to focus on two examples of viral reproduction. First, we're going to look at bacteriophage. Phage, remember, is the virus that infects bacteria. And then we'll also take a look at the viral life cycle of HIV. So we'll start with phage. And phage virus has this very particular and unique bacterial capsid. So they have the protein coat shell kind of up top here with the genetic material enclosed inside of it, and then it's got this whole additional part that's really specifically for attachment and entry. So we'll see that those parts become very important for the first parts of the viral reproductive life cycle or reproductive cycle of phage. So we'll start with attachment. So the phage virus is out in the environment, and here's my bacterial host cell. And what will happen is that the phage capsid will attach to the bacterial cell. So attachment is usually very specific, and so this is really what determines host range. So you and me, our human cells, we can be exposed to phage, but phage can't attach to human cells. It can only attach to bacterial cells, and so that's what limits phage virus to infecting bacterial cells. So there will be a very specific interaction between proteins on the virus and proteins on the surface of the bacterial cell. Entry is now when some part of the virus enters into the actual host cell. So in the case of phage, only the genetic material enters. This is not always the case. When we look at HIV, we'll see that the capsid and the genetic material enter. But again, in the case of phage, only the genetic material enters. So at this point, so we've got attachment, we've got entry, and then for phage, there's two options. It can either integrate or it can go directly to synthesis of viral components. So, and again, this is very phage specific. For other viruses, this will have to occur in a specific order. But for phage, it can go attachment, entry, and it can integrate, or it goes directly to synthesis of viral components. If it integrates, so the integration step, the viral protein, so a protein made by the virus called integrase, will cut open the bacterial DNA. And when it cuts open the bacterial DNA, it allows the viral DNA to enter. So in this image, the viral DNA is blue. It's now been integrated into the bacterial DNA. Once it's integrated into the bacterial DNA, it's called a prophage. And for phage, this puts it into a period of latency. So latency means a time when the virus is not actively reproducing, but it's still present in the host cell. So it's there, it's inside of the host cell, but it's not going to make new copies of the virus, and it's not going to kill the host cell. So for phages, integration is kind of a resting point. So that's the other way you could think of latency, as kind of a resting time for the virus. So what can later happen is that the prophage can come out. So it will actually no longer, it'll stop being integrated. And when it stops being integrated, that's when it can go into synthesis of viral components. So let me step back here for a minute. So remember this, I had the attachment, the entry. It can either integrate or go into synthesis of viral components. And what I just want to add on to here is that after it's integrated, that's kind of a resting or stopping point, but later on, it can go into synthesis of viral components. So it doesn't have to go through integration, but it can, and then later on go through synthesis of viral components. So for phage, integration is an optional resting period. We'll see when we look more closely at the HIV life cycle that HIV will have to integrate. So some viruses, integration is not an optional step, it's a required step. So once viruses go into synthesis of viral components, they're going to start using the viral DNA. So phage has inserted its viral DNA, and so it's going to use the host cell machinery. So it has to use the host cell DNA polymerase, the host cell RNA polymerase, the host cell ribosomes. It's going to take all of the host material and make the host make more viruses. So that's why I said it turns the host cell into a virus-making machine. So it will make copies of the viral DNA. It will also take viral DNA and transcribe it to make viral RNA, and then translate that to make viral proteins. So it's making more copies of the viral DNA, which we can see happening here, and then it's carrying out transcription and translation to make more of these viral proteins. So this is showing the capsid proteins. So as it's making lots and lots of these copies, then it's going to go into viral assembly. So in the case of phage, again, so specific to phage, it's going to take the viral DNA and mix it with viral proteins and assemble that into new viral particles. So that's what's happening here. We've got our capsid proteins, and I'll just draw them a little bigger here. So we've got our capsid, and we've packaged inside of it the viral DNA. And then the last step for phage is viral release. And in the case of phage, you can see that the viruses actually burst out of the host cell. And when they burst out of the host cell, it breaks open the host cell. So this is going to kill the host cell. Not all viruses are going to immediately kill the host cell like this. Again, we'll see when we look at HIV that it will bud out of the host cell. And when it buds out of the host cell, it will take away membrane. So we'll see that when we look at the HIV life cycle. So this is focused on viral latency and the phage choice that it has. So again, it goes attachment, entry, and then it can either integrate or it will go directly into synthesis of viral components. And then remember that it can also go from integration into synthesis of viral components. And then this is how we would make more viral particles. So these have distinct names for phage. So if it's integrated, it's called the lysogenic cycle. So let me use a different color here. So if a phage has integrated, it's just quietly resting inside of the bacterial cell, it's in the lysogenic cycle. If it is actively making new viral particles, it's called the lytic cycle. So let me circle these. This pathway is the lysogenic cycle. This pathway is the lytic cycle. And we can see that happening here. So we have, let's see, the attachment and entry of the bacterial chromosome. And so in the lysogenic cycle, that viral DNA becomes integrated. So it's now integrated into the bacterial chromosomes, and it's just quietly waiting there. Every time the bacteria divides, the virus gets copied, so any offspring of this original infected bacteria will be carrying the virus, but it's not actively making new viral particles. So no new viral particles, but again, it is getting passed to the offspring of this original cell. Conditions in the environment can cause an integrated virus to leave. So this is that integration going to synthesis of viral components. So the prophage can pop back out of the bacterial chromosome and then move into the lytic cycle. So once the DNA pops back out of the bacterial chromosome, now it will start making new viral DNA, new viral proteins. It will assemble new viral particles. Those will burst out of the infected cell and kill the host cell. And lytic cycle means to lyse or kill the host cell. So this is viral latency and phage, and again, these lysogenic and lytic cycle terms are specific to phage, but there's lots of examples of other viruses that have latency periods. Herpes is a classic example of this. So people who carry the herpes virus are always in, their cells always have it, they're just not actively making new viral particles. And so when somebody gets a cold sore, what's happened is the virus has come out of latency and is now actively making new viral particles. So it's only when somebody has a cold sore that they can actually spread the virus to other people, because again, that's the only time that new viral particles are being released by their cells. So we definitely see latency periods in lots of other viruses.